We're going to go over the rest of sensation and perception. So last week we covered chemosensation and somatosensation. So it covered things that we come in contact with, right? So like molecules that we either smell or taste, or physical touch. We'll get into today things revolving around remote sensation, so our ability to detect things outside of the organism. And then at the end of the lecture, we'll get into kind of more broadly how all of this information flows and creates perceptions. And that'll be kind of the low end of perception, and then we'll do object recognition on Monday that'll kind of build on that. So today's lecture, like I kind of hinted at, we're going to add on to last week's lecture, but today we're going to get into remote sensing. So this idea of detecting things that are outside of our body. And then we'll end the lecture getting into putting all of these senses together and creating holistic perceptions, both in higher areas for unimodal perception, so building on those individual senses, and then looking at early stages of multimodal perception, combining all of that information together. And we'll end with some cool studies that have looked at how we can reorganize areas that are lost due to people becoming blind or things like that. All right, so we are going to start with audition. So audition doesn't get nearly as much credit as vision. Vision is very widely studied in cognitive neuroscience. But audition is extremely complex and really, really cool. I mean, think about the fact that we have these really, really complex pressure waves hitting the ear. Think about being like in a crowded room or like at a party. You have so many sources of noise around you. You have music going, you have dozens of conversations. All of those sounds are all mingling together into these really complex waveforms. Yet somehow the brain is able to, first of all, turn this waveform that's in the air into a mechanical waveform in the bones in your ear, then turn it into a fluid wave in your cochlea, and then turn it into neural impulses, and somehow within this super complex wave of sounds is able to pick out a specific frequency so that you can hear your name all the way across the room among those dozens of conversations that are happening. Like, the precision of this system is incredible. So we'll get into kind of the individual pieces of it. Keep in mind, this is not a sensation and perception class, so we're going to kind of briefly go through these systems. There is a whole class on this. But the way that this works, kind of like I hinted at, you have sound waves in the air are pressure waves that are moving throughout the air. And these pressure waves enter the ear canal and they strike the tympanic membrane right here and cause it to vibrate, right? So you have this pressure wave in the air is now turning into a mechanical wave in the membrane, and it's causing these things to vibrate. And as this tympanic membrane starts to vibrate, it causes this chain reaction of vibrations to go down these three bones. Your ossicles, the malleus, the incus, and the stapes. And what ends up happening from here is that the vibrations that are mechanical at this point need to be transferred into the cochlea where the actual receptors are. And the cochlea is filled with fluid. And so this mechanical wave needs to be turned into a fluid wave. And there's this really kind of ingenious evolutionary mechanism. It has to do with surface area. The reason that the oval window is the size that it is because that bone needs to amplify the signal. Because if you have a signal that's vibrating through something like this, it's going to have to be turned up to be at the same amplitude going through fluid, because fluid will dampen the frequency. And so this bone, the stapes, is actually amping up the signal so that it's the same amplitude in the water that it was outside. So inside the cochlea, this is kind of a cross-section of the cochlea. We have these specialized receptors that we'll get into here in a minute, look at how they function. But they are the ones that are actually detecting the specific frequencies. And we'll get into how this is organized so that different areas of the cochlea are actually coding for different frequencies of sounds to create another map. The brain really likes to create maps of things. So inside the cochlea, it's this spiral-shaped structure, kind of looks like a seashell. And there's 3 fluid-filled canals in here. And as we just mentioned, so the stapes, this bone here, is the one that's transferring all of the vibrations into the fluid. And as the fluid moves through the liquid here, it starts to displace what's called the basilar membrane, which is here in the middle. And this is where the actual hair receptors are located that we'll talk about in a second. So as the wave is moving through this liquid, it's actually moving the basilar membrane as well. That's really important for what we're going to get into here in a minute. And the way that this is organized, so this is kind of an unwrapped cochlea. It's just been kind of stretched out. And you'll notice here that there are different frequencies of sounds labeled on here. And you'll also notice that as it gets closer to the end, it gets thinner and thinner and thinner. So it's that thickness of the basilar membrane that will vibrate at different frequencies of sound. And depending on which portion of the basilar membrane is vibrating, different neural signals will be sent, and the brain can kind of map out those different frequencies in the sound. So different frequencies are referred to as pitch. So how spread apart the different waves of the actual sound are determine what the note is that's being played. So really high notes are going to have really fast frequencies, high pitch is down here. Really low frequencies, so low pitch, low notes are going to be at the end of the cochlea. So main takeaway point here is that the ability to map out sounds is directly related to the thickness of the basilar membrane. So the hair cells themselves are pretty incredible, honestly. So we have inner hair cells, we have outer hair cells. They're embedded in the middle of the cochlea, in what they call the organ of Porti, here in the middle. The inner hair cells are the ones that are actually doing the sensation. They're the ones that are sending the neural impulses that the brain is interpreting. The outer hair cells, as we'll see in a minute, are serving to amplify the sound, to give the inner hair cells a better idea of what that frequency is. So they improve sensitivity. Also referred to as acuity, right? Our ability to distinguish sounds from one another. So the actual hair of these things, stereocilia, that kind of pokes up. So you have these little hairs right here, are attached to what's called the tectorial membrane. It's kind of this sheet that lies on top of the organ corti. And what happens, let's see, as the sound wave moves through the cochlea and starts to displace the basilar membrane that these are sitting on. So this is the basilar membrane right here. So the basilar membrane is waving, right? And as it waves, it pushes these hair cells up against the tectorial membrane, and it bends them. And this is not something I'm going to test you on. This is just kind of cool that we've kind of mapped out the molecular process that's going on here. It looks as cartoonish as this actually depicts. There's really this protein that's attached on one of these hair cells right here that looks like a coiled spring that's attached to this other hair cell right here. And as these hair cells get pushed up against the tectorial membrane and bend, this spring gets stretched and it pulls this gate open on the other hair cell. So it's a mechanical ion channel. And so it opens up and you have this flood of calcium and potassium that run down the hair cell that end up turning into the neural impulse. Like, that's incredible to me. Like, that is like a feat of evolutionary machinery that is absolutely insane. The outer hair cells, so these ones up here on top, they're also attached to the tectorial membrane. And as this basilar membrane is being moved and waved, they're actually adding more motion into the tectorial membrane so that it pushes down on the inner hair cells even more. And that's how it's amplifying the signal. So you have this wave coming through the basilar membrane, it's pushing these outer hair cells against it, which is pushing the tectorial membrane down on those inner hair cells even more. And it's making the signal greater. So when we talk about hearing loss, so our inability to hear loud sounds, that's damage to these outer hair cells. So there's no longer this amplification that's happening. So I want you to know that the inner hair cells are the ones that are actually doing the sensing down here. And the outer hair cells are responsible for giving those inner hair cells a better signal, or amplifying that signal. So the pathway, we talked about kind of in the second lecture about how the temporal lobe is responsible for audition, and it's kind of intuitive because it's right behind the ears. But you got to keep in mind that signals are not going straight to the temporal lobe. All of the sensation, except for olfaction, is going through these older brain structures first and then arriving in cortex. And so what happens first is that all of those hair cells, they all form the cochlear nerve that goes down into these hindbrain areas, and it first synapses in the medulla. This isn't stuff that I need you to necessarily memorize. This is just kind of get a conceptual idea of the flow of information, of how this is happening. And this is going to be something that comes up later, this point right here. So this colliculus right here that's down in the midbrain is actually really important for action when we get into action. And really early signals can be processed here to orient action signals. And so if there's a sound that's really important, then early processing before it even gets to the cortex can orient our eyes, can orient the head, and face us towards that thing so we can get a quicker reaction time to potentially dangerous stimuli. So the idea here is that sound is coming in through the ears, it's hitting those inner hair cells, those are going through these midbrain structures into the thalamus, and then finally out to the auditory cortex. And so there's a lot of individual processing that happens before it gets to what we actually consider auditory processing. And a lot of this early machinery is stuff that lower level mammals and other animals use for their types of auditory detection. This just allows for a much more flexible way of using that information, as we'll see throughout the course. So the auditory cortex itself, primary auditory cortex, this is where the information gets to first when it gets to the cortex, contains a tonotopic map. So I've hinted at this, that the brain really likes to create maps of the environment, maps of the body. We'll see that in vision as well. And so you see this really nice segregation of low frequency sounds, kind of more towards the anterior or front of the brain. And then you have these higher frequency sounds are organized closer to the posterior portion of the auditory cortex. And that allows it to create different profiles, because if it has everything mapped out, then it knows that when different things are on at the same time, that it's a specific type of sound. So that's what I was just getting at. They respond to very specific frequencies. And this is extremely sensitive. So they had some single cell recordings they were doing with epileptic patients. So they already had the brain open. They were able to get in there and do some recordings. A single cell would fire for 1,010 hertz, but not for 1,030 hertz. And that's a really small difference in frequency. So these cells are extremely specifically tuned. It's a very, very sensitive map of sound. But all of the other senses all have their own version of a map. You'll see the vision map is also extremely sensitive, in particular for specific spots in space. So just think of this, instead of being space, it's mapped out all of the available sounds, all of the options for sound. And you'll notice too that the frequencies listed here are the frequencies that are within our range of being able to hear. There's a lot higher frequencies and a lot lower frequencies, but we don't have detection power for those because they weren't important for our survival. So this is something I'm going to explore in this next slide here, but this tonotopic map is preserved as the information flows to higher processing regions. So you have auditory cortex is getting the raw signals, and then those are being kind of combined and associated in this A2, where you still have some type of tonotopic map, and I'll show you that on this next slide. So important thing to know here is that in both auditory cortex or cortices, so in primary auditory cortex and in secondary, there is tonotopic organization. And this was a study that actually mapped that out. So they were using a 7 Tesla scanner, which is twice as strong as the one that we have here. So they're able to get a lot better resolution and focus on things that are much smaller than other MRI imaging modalities are able to get you. And so they were basically just delivering different frequencies of sound to subjects while they were in the scanner. And then they just mapped out where the brain was lighting up for those individual sounds. So it's not perfect, you'll see here, but you do have kind of lower frequency down here, and it gradually gets to higher frequency up here. And so this was a pretty well-preserved finding. Like the researchers in neuroscience were pretty convinced that there was this really strict kind of pathway of information where the entire map was preserved in A2. But what they started to find with mice was that if you look down at the microscopic level, even though there's this macroscopic organization of tone, where you can see there's very specific regions for like low frequency and for high frequency, that when you look down at the individual level, there was some variability within the individual cells. And so this is kind of, these plots right here show that like, even though there's kind of a gross like preference for a certain type of frequency, that the individual cells around that frequency were responding to a bunch of different ones. And the idea here is that, or what they think is going on, as we'll see when we get into perception and into object recognition and things, as information flows from these primary cortices to secondary and higher association areas, it's trying to identify what those sounds are and where those sounds are. And so what they think is happening here is that in secondary auditory cortex, there's actually identification happening. And so a certain sound may usually have a low frequency. Let's say it's a sound for a particular predator or for some type of prey. But that animal also makes other sounds. And so they think that there's kind of a general organization of the sounds that that animal usually makes, but then there are other cells around it that help specify those other individual sounds that are kind of associated with it. So the goals of the auditory system, this is something I was just kind of hinting at. So there's two main things that these higher areas are interested in, and that's what type of object it is, what type of sound it is, and where is that sound in space, right? And we're talking about remote sensation. Like these are goals for survival, for finding resources. We want to identify things, and we want to know where they are in relation to where we are. And the auditory system has very specific mechanisms for both of these goals. So it has identification type features, and it also has features for locating things. Intraoral timing is something we'll talk about here in a second. Really, really cool mechanism of the auditory system. So with identifying sounds, the auditory system uses something called timbre, which we'll talk about here in a minute. Timbre is kind of the body of the sound, the texture of the sound. Even though a G might be played from a guitar and a viola, you can tell that that G came from two different instruments. It has kind of a unique profile. And that's what we'll talk about here on this next one. So these are ideas that we're going to come back to on these next couple of slides. So don't worry if you don't get all of this down. So we're going to talk about timbre in a second. We're going to talk about intraoral timing next. Main idea is that what and where are very common things that are going to come up throughout the course. Those are goals that the brain has in general. So timbre. So sounds are composed of different properties. So you have pitch, which we've talked about, which is the actual frequency. And so a tuning fork delivers pure pitch. There's no like resonant frequencies, which is what you get with these other instruments, because as the sound goes through whatever the shape is, it resonates and it creates these like different wave patterns that become kind of the identifier for it. And so this is kind of what I was getting at, that even though you have the letter G being played from these two different instruments that are the exact same pitch, we're able to distinguish the difference. And it's because they have this kind of profile, this wavelength profile that we're able to pick up on. So every object, every living thing does make sounds with its own resonant frequency. So when we talk, you can actually change the resonant frequency of your voice by changing the shape of your mouth. by moving where your tongue is, by making your mouth bigger or smaller. Those are what give your voice its identifying feature. You have your own timbre. Location is a really, really cool feature because this is happening within milliseconds. And so the phrase is interoral timing. That's a term I want you guys to know. And the idea behind it is that when we hear a sound, our ears are a certain distance away from one another, right? And so if the sound's coming from over here, it's going to hit this ear before it hits this ear. And we have these coincidence detectors in our brain that will kind of measure the time delay between how long it gets from one ear to the other and can orient associated to that. Like, that's freaking incredible. I mean, that's like a millisecond difference for it to get to one ear to the other. We don't consciously perceive that, we're not aware of that, but somehow the brain is able to do this calculation and is able to locate, okay, the sound came from the right, the sound came from the left. The shape of our ear also makes it so that we can do up and down location. Because if it's coming from the top, then it's going to bounce off of this part of our ear, this part of our pinna, and then bounce up into the ear. And so this one's a lot more complicated. I don't know the nuance of like how the brain is actually calculating that stuff, but the shapes of our ears are very particular for locating sounds. A lot of the work that's been done with interoral timing has been done with like owls. There's actually a lot of people here that are doing that kind of stuff. This is something that's really tough to study in humans because the timing is so small. Like when we're doing MRI, we're looking at like 5 to 12 seconds. So this isn't something we can reliably pick up on. And then if we're looking at it with EEG, it's really hard to locate it to a specific region. But there are a lot of people that think this is happening really early on. And the reason they think that this is happening really early on is because of the orienting reflexes, right? Once we know where a sound is coming from, before we're consciously aware, we're already turning our head to look at it, right? So interoral timing, difference between when the sound hits one ear and the other. Cool. So that is end of Audition for now. There's a lot more to Audition. I really recommend you take a sensation and perception class. Really cool stuff. But we're going to start getting into vision, which is also extremely complex. Like we have these photons that are bouncing all over the place, and some things absorb photons, some things have photons bounce off of them. That's what gives them their color. You'll see in a second the reason that the pupil is black is because the retina absorbs all of the light that goes into it. And so it appears black because there's no light bouncing back out of it. So getting into the structure of the eye, because the eye is one of, apart from the ears, is one of the most amazing evolutionary feats. So the outside of the eye is the cornea. It's kind of this convex covering of the eye. This portion, it's composed of cells. It's composed of living things that need resources, they need oxygen to survive, but you can't put blood vessels there because it'll block the light. And so the way that the cells of the cornea get replenished is that they absorb oxygen from your tears. And that's why the eyes have to constantly be lubricated. So the cornea does have a function within like the focusing of light, but that focus is fixed. So the cornea doesn't really change shape throughout your life unless there's some kind of disorder or disease. So however light comes through the cornea is going to be the same no matter what's going on in the other parts of the eye. The iris is the colored portion. It is a circular muscle that is within the cornea. And these circular muscles can retract or they can, when they contract, they get bigger and the pupil opens up. And then when they relax, it closes this circle and the pupil gets smaller. So the pupil is actually just a hole. It's not an actual thing. It's just what the iris is doing, and that's allowing either more or less light to come into the eye. And the iris itself contains melanin pigments, And that was intended to try to filter light to make it so that light wasn't coming through the iris, because the only light that was supposed to come through was through the pupil. So early in evolution, everybody had brown eyes. And then there was a genetic mutation that happened on one of the splits in Russia and in Europe that ended up with green and blue and all the different color eyes that we see today. The main function of that melanin is to distort the light so it's not coming through this area. Yeah. So behind the iris is the lens, and this is where a lot of the magic happens for focusing things. So it's a biconvex crystalline structure that's surrounded by these ciliary muscles. So the iris is not what's stretching out the lens. There are specific muscles that surround this thing, and the ciliary muscles that are able to change the shape of it. And so when the lens is constricted, it's round, and you're able to see things that are closer to you. So that's kind of this picture down here. And then when it's stretched and flattened out, it makes it so that you can see things that are farther away. And this is happening extremely fast. I mean, if you are looking at something close and then look at something far away, it's doing that calculation really, really fast and changing the shape of that lens. So when it's relaxed, you see things that are closer, the lens is round. When it's stretched out, it flattens things out, and it spreads out this field of view so that you can see things that are farther away. And this is something I always like to show my participants when they do brain scans. This is my brain, but you can see the crystalline lenses in the eyes right here pop out really well on MRI. So the retina, there are a lot of people that argue that this couldn't have happened through evolution because the eye is so complex. But when you really look at the structure of the eye, it's kind of built backwards. It's kind of inefficient. The cells that are actually receiving the light are all the way in the back. So light is focused by the lens on the back of the retina, which is this whole portion of the back of the eye. And it's made up of 3 layers. And so the ones that are actually doing the detection are all the way here in the back. So the light's actually got to make it through these top 2 layers to even get to the ones that are detecting the light. They then pass their signals on to bipolar neurons, which are here in the middle. And bipolar neurons then activate these ganglion neurons, and those are the ones that are sending the signal to the brain. Those are the ones that form the optic nerve. And we'll see some properties of those here in just a second. So this spot where they leave is the optic nerve, and when you're getting an eye exam, they call it the optic disc. There's this big circle in the back of the eye. It's a blind spot. There's no photoreceptors in that part of the eye. There's some cool tricks. If you look on YouTube, you can draw an X on a paper, and you can put it so that it falls right in your blind spot, and the X disappears. And so our brain fills that information in. It makes stuff up. It uses the context around that blind spot to make guesses about what should be in that blind spot, and it fills all that information in. Your brain makes a lot of stuff up. So we'll get into these different components here on these next couple of slides. So the photoreceptors themselves, so we have rods, which are more on the outside, the periphery of the retina. So that's why there's a ton of rods out here, a ton of rods out here. Not a lot of cones. The cones are the ones that are more in the center. So there's a ton of cones right here in the fovea, and not a lot kind of out on the edges. So rods are the ones that are sensitive to low light, and we'll see why in a minute. And then the cones are the ones that are picking up color and are really sensitive to bright light. And they make up the fovea. The fovea is the most important part of the eye for our perceptual abilities. There's different kinds of cones that detect different types of frequencies of light. So there are certain cones that are specific to blue light. There are certain ones that are specific to green light, red light, and all of the colors that we see is a mixture of those. So mantis shrimp have I think 16 different photoreceptors. They can see crazy amount of colors. Just saw some video where they put 3D glasses on a mantis shrimp. And it actually thought things were coming at it. It was crazy. So I'm sure this is pretty much a review for you guys. It should be. So something to keep in mind with this portion right here, I want to show you on this last picture. You see this tiny little dip right here? This is the fovea. This is how small the fovea is compared to the rest of the eye. So there's a ton of cones packed into that tiny little spot right there. And this is not something I'm going to test you on. This is just some cool kind of molecular machinery that's going on under the hood. So these photoreceptors, they have a molecule in them called opsin that will break up when light hits it. The cool thing about these, though, is that it's opposite of what you think. So these are constantly firing when they're in the dark, called dark current. And the breaking up of the opsin actually closes the ion pores and stops it from firing. So these photoreceptors, they're constantly releasing an inhibitory neurotransmitter that is causing these bipolar cells not to fire. And then when light hits it, the inhibitory neurotransmitters stop and this thing starts to fire. And so light is actually doing kind of the opposite of what you'd think it is doing. But then when light hits it, these bipolar cells are then sending signals to the ones that are sending signals to the brain. So that's dark current, if you ever hear of it. Not something I'm going to test you on, but kind of cool background knowledge. And that's what I was just talking about. So the opsin is actually what's closing the pores, not opening them. So this is where a lot of the acuity happens. So we have 260 million photoreceptors, and we only have 2 million ganglion neurons. So we have a ton of cells that are picking up light, but all of that light is being compressed, right? So, and a lot of it, so a huge amount of rods will activate one ganglion neuron. So it's a summation principle. And this is what allows us to see in low light, is that these ganglion neurons are actually summing up a ton of rods to try to get an accurate representation of how much light is hitting the eye. But on the flip side, you can have one cone activating one ganglion neuron. So it kind of shows you the disproportionate representation of cones to rods. The actual signals that are going to the brain are much more coming from the cones than they are from the rods. And this is what allows us to see the detail that we are. You can kind of see that here. This is a very simple representation of it, but it kind of makes the point that like this one cone is activating this one ganglion neuron, and here you have multiple rods that are activating one ganglion neuron. So a lot of the real estate is devoted to the cones and to the fovea. And like I said, the fovea is this tiny little divot here. A really, really small part of the retina. But that's where a ton of our acuity is coming from. So just know that few rods per ganglion, or lots of rods per ganglion, few cones per ganglion. And that allows for finer detail. So getting into the fovea. So it's located in the center of the retina. Like we've been talking about, it's responsible for our ability to distinguish between different items. And this is all kind of repeat what I just said. So this is kind of building on what I was just talking about here. So half of the optic nerve contains information from just the fovea, which is huge. I mean, you have this whole portion of the retina, and over half of the information going to the brain is coming from that one tiny little divot right there. And the whole purpose of moving your eyes, of saccades, eye movements, is to focus light on that tiny little spot. And that's happening non-consciously. Frontal eye fields is something we'll talk about when we get into action. Those are what are controlling eye movements unconsciously, and they're actively involved in attention mechanisms and things like that. So really big part of vision is trying to focus things on that tiny little dot right there. And this has come up quite a bit. I guarantee you there's going to be an exam question about the fovea and acuity. All right, so left and right visual fields. So most people kind of have this intuitive sense that the left eye goes to the right half of the brain, the right eye goes to the left half of the brain. It's not like that. So the left and right visual fields are actually coming from halves of the eye. So The left visual field is coming from the nasal portion of the left eye and the lateral portion of the right eye. And then the right visual field is coming from the lateral portion of the left eye and the nasal portion of the right eye. So each visual field projects to an opposite side of the brain. Oh, you can kind of see it on here. So so let's say like this spot right here is where the optic nerve is leaving. This half of the eye is coming out and forming one optic nerve, and this half of the eye is coming out and forming a different one. And so there's actually two wires coming out from either side of the eye, and then those wires are then crossing to the other side of the brain. And so you see that this one, so for the left visual field, it stays on the right side of the brain, whereas this part of the left visual field from the left eye crosses over into the right side of the brain.
Is this common for like other animals?
Mm-hmm. Yep, and it's mainly built on the fact that the optic nerve leaves kind of in the middle of the eye. So this will be really important when we look at the way that the visual cortex is organized, because it separates out information from the left and right eyes. All right, so big idea from here is that the left and right visual field are not coming from separate eyes, they're coming from halves of each eye. I don't necessarily expect you to remember exactly which one is coming from where or anything, just kind of know that the intuitive distinction is not what's actually happening. So the visual pathway, so this is something I don't need you guys to remember, but it's a really interesting piece of information that will kind of provide context for future lectures, in that vision is given a lot more weight in perception than other sensory modalities are. So 90% of the axons take the normal pathway to the visual cortex. It's called the retinogeniculate pathway. They kind of go through the LGN in the thalamus and cross the optic chiasm and then end up way back here in V1. But there's this whole other path, 10% of the axons take this shorter path that go to midbrain areas. And this is for like orienting movements, for moving our head and moving our eyes and things like that. What I want you to take from here is that this 10% is more fibers than the entire auditory tract. So 90% is way more than, so like this path has a ton of information compared to our other senses. And that 10% that's just used for orienting motions, moving our head and moving our eyes, is still a ton of information. So I don't need you guys to remember like all of these different brain regions and things like that. Just know that there's a lot of processing power devoted to vision. So receptive field. So this is getting into how the information is being built on and how it's being kind of coalesced and organized at higher and higher levels. So as we get to these areas of processing in the visual areas of the brain, we have these cells that have very specific receptive fields. So this is the LGN, the lateral geniculate nucleus, in the thalamus. And the cells here that receive visual input respond to spots of light in very specific locations. And so this bar will light it if it kind of lands right there. If you shine a light just on that tiny little spot, it'll activate it like crazy. I don't need you to know kind of the nuance of the off and on. It's basically this center off surround. So any kind of light that hits the outer perimeter is going to inhibit activity, and any light that hits the inner part is going to activate it. And so the fact that the middle is completely covered in this one means that the cell is really highly active. What I want you to get from this is as we move from the thalamus to the visual cortex, which is right here, you start to see that these simple receptive fields are being combined into more complex receptive fields. So these three different cells are being combined into one that will respond to a specific orientation. And so in the thalamus, you have cells responding to very small, specific spots of light in the visual field. And by the time you get to visual cortex, the cells are actually firing for lines that are at specific orientations. And this allows us to then build objects. Because if we have a horizontal line and we have two vertical lines, it's probably a table. And that's how things are being kind of built upon from this early information up to higher order information. And this was actually an accident, the way that they found this out. They were studying these LGN neurons that they knew would respond to these specific spots of light, and they started recording from V1 neurons in a cat. And they were showing these spots of light in all these different places, and it was really finicky. It wasn't lighting up the right way. And then all of a sudden, he accidentally moved the whole slide across the screen, and the orientation of the edge of the slide was what ended up lighting these ones off. And so they started to realize, like, okay, these cells are responding to lines instead of spots, and that's allowing for the building of complex perception. So that's where we get into here. So the visual cortex has a retinotopic map. So we talked about tonotopic maps. These are maps that are very specific to where on the retina light is hit. And so if this is our entire visual field up here, these are the regions of the brain that are processing those portions of the visual field. So that's what we mean by retinotopic. You'll notice that more cortical surface area is devoted to foveal portions. So you have this middle portion right here, 1, 2, 3, and 4, have really big representation here on the brain compared to these more peripheral areas. So like 9, 10, 11, and 12 have really small areas, and that's kind of our main periphery stuff that's coming from a lot of the rods. So this is just like when we were talking about somatosensation. If we use it a lot more, then we have a lot more cortical real estate devoted to it. So this is where the distinction about the visual fields is really important, because this left side of the brain isn't just receiving input from the right eye, it's receiving input from the left and the right eye. And so what the brain is doing is it's creating these ocular dominance columns. And so there's an entire column of cells that responds to the left eye in that specific spot of space, and then to the right eye, and they alternate. So there's left, right. They're all coordinating to a specific spot in space, and each of these columns is also specific for a different orientation of a line. So horizontal, crooked, vertical, whatever. You kind of see as we go along those columns that there's different orientations. And we'll see in the next slide how they actually map that out with MRI. And this is kind of that graph too, of showing these cells different orientations and high firing rates for orientations at a specific angle. But the reason that it's called ocular dominance columns is because each of these columns is from a different eye. So this is going to kind of touch on this. I wish I could spend more time on visual areas, but we have a lot to get through in this class. So as you move from V1, these primary visual areas, into these higher processing areas, so secondary visual cortex, into some of these other visual areas, your receptive fields become more and more complex. And by receptive field, I mean what you have to show to that cell to get it to fire, right? So in LGN, we were showing really small spots of light, which were a 1 degree visual angle. So these are specific to a very, very small spot of space. V1 neurons are about twice as much space. So they take up about a 2 degree visual angle. Some neurons, as you get to these higher processing areas, some neurons will respond to an entire hemifield of the eye. So they become really complex. Some of them are tuned to motion, some of them are tuned to shape, color. You'll notice there are ones at the end that will light up specifically for a face or for a hand. So we have these really simple orientations, these really simple lines that are being combined into complex features, so that cells near the higher ends of the hierarchy are lighting up for very specific types of features and for complex objects. And so that's kind of what I wanted you guys to get away with, the flow of information that I was talking about at the beginning of the class, is that this is the building process. This is how our perceptions are created from the ground up. You have these really simple sensory signals that are kind of being combined. That's why they're called association areas, because they're associating all of these simple features together into complex objects as it moves down. There are 30 distinct visual areas in monkeys, so they've done a ton of visual work with monkeys. They believe that we have even more in humans because we have a lot more recognition abilities than monkeys do for very complex objects. And this kind of gets back to our very first lecture where we were talking about early neuroscience, where they're trying to make the distinction between localization and aggregate field. So is the whole brain involved, or are certain functions localizable to small regions of space? Phrenology was way off base by saying that like these really complex cognitive things were localizable. What we're seeing here is that there's definitely localization. There are specific regions of the brain that are processing very specific types of information, but it's not these higher cognitive combativeness, stuff like that. It's a building process. So this was MRI being used to map out these ocular dominance columns. So, this was mapping out the retinotopic map. That's on the next slide. So in order to look at what regions of the visual cortex were lighting up for different places in space, they're able to do two types of studies. So they either do a rotating stimuli, where they have this kind of checkerboard pizza pie looking thing that will spin, and as it spins, it's lighting up different areas of the visual field. And they can map that out on the visual cortex. So this color wheel is kind of showing different spots in space, and it's showing which areas of the brain were represented by those different areas of space. It's not a perfect map, but it's really well segregated. And then the other one was a dilating stimulus, so it was able to get really small and then really big. And so they used this to try to map out which areas were devoted to foveal vision versus peripheral vision. And so these areas in red are foveal, and then as you get further out, it's more peripheral stuff. Don't read too much into like where the brain regions are and things like that. These are just methods that are being used to try to look at this at a macroscopic level with MRI. And then this was the really cool one. So they used a 7T scanner. So this is, like I said earlier, a really powerful scanner. And they were looking specifically at V1. So this is just a chunk of V1. And the blue and red is indicating whether or not the information came from the right eye or the left eye. And so they were able to see with an MRI, which these are like thousands of cells within each of these voxels right here, but really cool organization here. And then they were also able to map out the orientation tuning. And so these are all the different line angles that these different cells were responding to as well. So you can see really clear segregation of all of these types of information from different parts of the eye relating to different features within the stimuli themselves. This is a really, really powerful study that demonstrated this at the molecular level. And this was done in humans because this is important. A lot of studies in sensory modalities are done on animal models. A lot of what we know is done in cats, is done in monkeys. And so the ability now to scan at really high resolution is showing that some of this stuff can translate to humans, because that's a big thing in research. Like the neuroscience faculty is doing amazing work with rats and mice, but it's like how much of that is translatable. There's an entire Twitter feed that's just called In Mice that finds popular science news articles and then reposts them with the headline In Mice, because they're like, oh, did you know that diet does all of these things? And they're like, in mice? So this is a really cool demonstration that we're starting to translate this to humans. So is the structure hierarchical? So evidence that it's a purely hierarchical system would mean that all of the information is being preserved in each area of processing, right? And we see a lot of evidence for this at these early areas. So from LGN to V1, we see this really nice preservation of the individual spots of light and all that kind of stuff. But as we get to higher stages of processing that we'll get into in the next couple of lectures and a little bit more in this lecture, you start to see that the brain with higher cognitive stuff is using more of a divide and conquer strategy, where it's not preserving all of the information. You have a specific region that's looking at color, you have a specific region that's looking at motion. This is where I was talking about, like, localization does happen. There are local regions that do specific things. And it does separate out the information a little bit, because certain areas of the brain are more interested in whether or not a thing is moving, and certain areas of the brain are more interested in whether or not it's the right color food that we can eat, right? You have certain parts of the brain that need to respond to movement, need to respond to threats to the individual. And so it's able to kind of segregate out all of this information at higher levels of processing. And this is a really nice demonstration of that from MRI. And so they showed subjects either this collage of colored blocks or a collage of just black and white blocks. And then they also, with this kind of collage of black and white dots, they either showed them a stationary one or they showed them one where all of these blocks were kind of moving around in space. And what they found was that there were very different regions responding to the motion condition than were responding to the color condition. And it kind of demonstrated this segregation really well. So these early visual areas were active for both. So this is early visual cortex. But as the information started to be processed from the early visual information, it got sent to different regions depending on whether it was coding for color up here or it was coding for motion down here. And so this kind of demonstrates that divide and conquer strategy with MRI. This makes it tough to see exactly what type of motion or what type of color. This was a PET imaging study, so the resolution's not really good, like that 7T scanner one that we just looked at. But this is still a really good example of it. So this is the end of vision. And so what I want you guys to kind of take out of this is this idea I keep coming back to, the flow of information. The idea that all of this sensory processing is a building process, right? Our actual perceptions are a lot more complicated than what our sensory systems are picking up on. And that gets us into perception. So, our ability to identify things, like we talked about this in our lecture last time, where let's say you smell something and you have these molecules that are hitting the receptors in your nose, and it's setting off this profile of neural responses, and then somehow you're able to say that was a grapefruit. And not only are you able to say that was a grapefruit, but you're able to picture what a grapefruit looks like, and you're able to imagine what that grapefruit feels like and what it tastes like. Those are all high order perceptions that were based on really simple molecules that hit your nose. So this is that, again, what I keep hitting on, this idea of how the information is flowing. So we've now that we've gotten to the end of sensation, we've seen that all of these different senses have these primary cortices where the initial processing is happening. So you have auditory, you have visual, somatosensory. And then as these simple features get more complex, they get spread out to these secondary cortices that are bordering the primary ones. That's really important because they're actually getting synapses from the primary cortex. They're consolidating information, adding more to it, associating different information from different parts of the primary visual cortex to identify objects. And that's kind of what I was getting at here. So they're trying to figure out what the items are and where they are in space. I mean, this is the most important thing for survival, right? We need to identify food. We need to identify threats. We need to know where the food is and where it is in relation to us, where the threat is in relation to us so we can protect ourselves. And then as we'll get into here in just a minute, these unimodal association areas then feed to much more complicated multimodal areas where you have sensory cells that can be activated by more than one sense. The frontal lobe is a huge integrated processing center. That's why a lot of theories are out there about it being the control center, because it has so much information from so many different places. And the multimodal stuff is what allows us to do what I was just kind of getting at, taking that smell and turning it into this whole perception of bringing in visual information and bringing in taste information and all that kind of stuff. Like I can sit here and just walk you through a meditation where you think you're actually biting into one, taste it, see it, And all of that is happening because we're taking information from these early sensory areas and we're combining them together in these multimodal areas. So where is perception? So there's a lot of evidence that it's occurring at the higher processing areas. So what we actually experience, what we feel, tends to be coming from these higher areas and not necessarily the lower areas, the primary areas. Some evidence comes from this illusion. So if you stare at the middle of this, you'll notice movement in the blue rings. Your primary visual cortex is not identifying any actual things in those blue rings. The percept is linked to the motion areas, and they've seen that on fMRI and PET. That like, even though there's nothing moving here, we perceive it, and the areas that calculate motion are active for illusions like this. So this is evidence that what we experience is not necessarily what we see. It's not increasing any activity in the primary regions. So this kind of gets back to some scary ideas if we're talking about brain-computer interfacing, because we can actually drive perception. And so they had these monkeys and they were training them on this task where they had to make an eye movement towards the moving direction of these dots. So these dots would move in a certain direction. The monkeys were trained that they needed to make a saccade, an eye movement in the direction of those moving dots. And so they did this test with these monkeys for quite a while, and while they were doing it, they were finding which cells were responsive to specific directions of motion. So they identified the cells that were specific to this upward right motion. They found ones that were specific to an upward left motion. And then they stimulated those while they were doing this task. And instead of doing an eye movement towards where the dots were actually moving, they did an eye movement towards the direction that was being stimulated. Now, we can't ask these monkeys whether they saw the dots moving in the wrong direction, but their behavior is definitely showing us that their perception was warped, right? So we can drive these processes. We can trick the brain by giving it false signals. Kind of freaky. So that was kind of what I was just getting at. So they moved in the wrong direction. So this is something we can manipulate. So multimodal. So these are highlighting regions that we've been talking about that are either audio-visual. These sections up in the frontal lobe are trimodal. So they can take in auditory, visual, tactile information. And so the actual neurons are firing to multiple signals. And these cells actually have a stronger activation when multiple signals from different senses actually converge at the same time. And so the brain actually gives a lot more weight to multimodal signals than it does to unimodal signals, because there's more evidence of that stimuli with a multimodal representation just from one of the senses. So this idea of convergence is important. It's kind of a coincidence detector. It's relying on the fact that all of these things are happening in the same location and at the same time. And it's kind of crazy that the brain's able to do this when you really think about it, because sound information and touch information and sight information travel at different speeds. And somehow, like, it's able to delay some while it waits for others. Like, perception is actually believed to be a lag. So we're actually seeing things after they actually happened, because our brain is like slowing them down to take in the rest of the information. And so I'm going to talk about this convergence idea on this next slide. So these multimodal activations require that whatever's happening in the outside reality is happening in the same time and in the same location. And you can do this, you can look this up online, it's really cool. It's an auditory driving illusion. So if you see one flash of light at the same time that you hear two beeps, you'll actually see two flashes of light. Your brain is like, those things are converging. If they're coming from the exact same location and they're happening at the exact same time, your brain builds up the representation that those things must have been connected with one another, and you'll actually see two lights. If they're presented from different locations, or if there's even just a slight millisecond delay between the sound and the light, then the convergence doesn't happen, and we don't see two flashes of light. Another bit of evidence that our perceptions can be tampered with and altered. And it's because of the fact that these multimodal cells are, because a light signal and an auditory signal hit that cell at the same time, it became really active and said, this thing is the same thing. And so it was like, okay, if there's two beeps, there has to be two lights. And it happens, it's really quick. Like the beeps are really fast. This was really cool though. So they did this study with lip reading, where they showed videos and had sound hooked up. And there was this region of the brain called the STS, which is a multimodal region. And it was active when the sound and the recording were in sync with one another. So when they were actually like reading the lips and hearing the sound. But if you separated the sound, and you guys have seen those movies or like the dubbed movies or whatever, this region wasn't active. So it was converging the fact that the video and the sound were the same thing. But once you separate in time the sound and the video, this multimodal region stops working. And you have this kind of sense of like, this is weird when you're watching movies like that where it's not lining up. So, yeah. So the McGurk effect. So this is just the idea that visual signals are more dominant, right? So more reliable. And I'm going to go through this pretty quickly. This was, if you hear someone say ba, but you see a video of someone saying ba, you'll actually report hearing what you saw versus what you heard. So the brain is actually giving a lot more credence to the visual signal than it is to the auditory signal. And you can kind of trip people up on this. So I got some cool studies that I want to show you here at the end. So I'm going to skip through. So synesthesia is a really interesting disease. It's kind of this faulty wiring. It's believed to be kind of a disruption of these multimodal signals, where people will actually report seeing color for specific words or specific sounds and things like that. And they did this study. I'll show you some limitations of it. I'm not buying it completely, but they took word color synesthetes, the ones I was just talking about, and they had them listen to pure tones into words. And so the pure tones, they didn't report seeing any sound, but they are seeing any color. But the synesthetes, for specific words, reported seeing very specific colors. And so they scanned them and related them to controls, and they actually found that these multimodal areas and visual processing areas were lighting up in these synesthetes that weren't lighting up in controls. And so it was like this evidence that those brain regions are actually coming online. It wasn't, they didn't find a ton of overlap for the actual color reported and the one that they were seeing, which I don't know how much you want to read into that because this had 13 synesthetes, not a huge study, and 28 controls. So really cool that they found these multimodal areas and things like that were lighting up, but it was definitely an underpowered study, like a lot of these early ones are. This book mentions a lot of early fMRI studies. There wasn't a lot of grant funding in the early 2000s, and so they did a lot of studies with like 30 people. This was more evidence from diffusion. So it's a different type of synesthete. They looked at just white matter and found that overall synesthetes had a lot more white matter connections. And especially in this certain area, this inferior temporal gyrus, they claim, I don't really buy this one either, it's really cool that they had this finding. They say that the amount of white matter in this area was able to predict whether or not those people actually saw color in the outside world or just saw it in their head. There were only 18 people doing group different stuff because like there were a small number of these 18 that saw things out in the world, a small number that saw them in their head. Comparing groups like that for things like that, this isn't the best way to do science. But this is an interesting finding that there's more connections, and that might be why there's some cross-wiring in these multimodal regions. So I'm going to run out of time. There's perceptual reorganization stuff. I was going to show you about, this is a really sad slide, so we can skip it, because it's about babies losing sight if they have cataracts when they're young. But this is the one that I wanted to show you before we left, because this is really cool. Blind individuals, they had them swipe their fingers over bumps in a tactile discrimination task, and then they had them do it for actual Braille reading. And they found that in the blind individuals, while they were doing the Braille reading, the visual cortex had lit up. And they also did another one just like it, where they used auditory stimuli, and they were telling them to distinguish between two sounds. Visual cortex lit up for this too. And so this is evidence that when you have takeover of these regions, it's usually multimodal. It's multiple senses converging in these areas. So that's what we'll end for today. That's some other stuff. This, if you guys have this video down here by David Eagleman is really, really cool. So check it out.